So here's another view of the transcriptional apparatus. You see that here's a generic gene, and this generic gene is going to have a promoter sequence and then a coding sequence. And of course, this gene is a double-stranded DNA entity, and so one of these strands is going to be a coding strand, and the other strand is the template strand. The template strand is going to be read by the RNA polymerase from the 3' to 5' end, and this RNA polymerase is going to look for the promoter sequence, and it will come in, start reading the template strand from 3' to 5', and thus synthesize an RNA from 5' to 3'. Let's take a look at that again. Think of the promoter as the landing lights. There's an airstrip and an airplane. The RNA polymerase has to come in and land at the appropriate place on the airstrip. Well, these are like the landing lights on that airstrip. They're going to guide the RNA polymerase in. It begins transcribing at the +1 sequence. It's going to read the template strand from 3' to 5' and synthesize an RNA product from 5' to 3'. Remember that. Tuck that fact away. RNA is transcribed from 5' to 3', just like DNA was. It hasn't changed. Now, the promoter sequence itself, remember that it precedes the gene. It's the binding site for that RNA polymerase coming in. It's like the landing lights. It's like a beacon. Boo, boo. It's going to guide the RNA polymerase in so it starts at the appropriate place. It's a double-stranded sequence, the promoter is, but only one of those strands is read by the RNA polymerase. A couple of other terms to define here. You see this upstream and downstream. The upstream simply refers to anything that's typically towards the 5' end of the coding strand of DNA. So we'll use the coding strand as the sort of our baseline, and anything towards the 5' end of that coding strand is said to be upstream. Anything towards the 3' end of that coding strand is said to be downstream. So if we use the promoter as sort of the reference point, anything that's closer to the 5' end of the coding region is said to be upstream of that promoter. Anything closer to the 3' end of that coding strand is said to be downstream of the promoter. This will have some implications later on when we talk about upstream enhancer sequences, downstream enhancer sequences, upstream promoter sequences, etc. So we'll come back to that theme, but I want to define it for you now. So shown on this particular slide is the way that the genetic information would flow. A couple of key points that you need to realize from this slide are that, first of all, DNA is double-stranded, and there's going to be, for transcription, one of those strands that's a coding strand, and the other that's going to be the template strand. We've already mentioned that the template strand, you're going to recognize this because in the direction of transcription, the template strand is going to be the one that reads from 3' to 5'. The RNA polymerase then would come along, it would look for promoter sequences on both the coding and template strand. It would then, at the +1 site, would initiate transcription by reading the sequence on the template strand and synthesizing complementary anti-parallel sequence where it was reading along the template strand. So along the template strand, notice this read from 3' to 5', read TACC, etc., 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 CTG. Well, the RNA polymerase would then put in, where it saw a T, it would put in an A. Where it saw an A, it would put in a U. Where it saw a C, it would put in a G. On and on and on and on, till finally at the end, where it saw a T, it would put in an A. Where it saw a G, it would put in a C. So in the end, after the transcription occurred and termination stopped transcription, you would see that this sequence reads in a complementary anti-parallel fashion to the template strand. Okay, so the 3' end of the template now base pairs with the 5' end of the mRNA, going all the way to the 3' end of the mRNA, which is base pairing with the 5' end of the template strand. Let's take a step back now and compare the sequence of the mRNA that's been produced with the coding strand. And notice that they're virtually identical. The only difference in sequence is that where there was T's in the DNA, there's now going to be U's in the mRNA. Where there was a T here, there's now a U. So the coding sequence of DNA reads exactly like the messenger RNA from 5' to 3', except the T's are replaced with U's. Okay, now if you're assuming that you're in a eukaryotic cell, this messenger RNA is going to leave the nucleus, go out into the cytoplasm, and it's going to be bound by a ribosome, which will now translate this mRNA information into protein sequence. In the prokaryotic cell, there's no separate nucleus. So as soon as the mRNA is being made, a ribosome is going to hop on board and start translating this into protein information. Now, what do I mean? How is this accomplished? Shown here in the messenger RNA are these 3 base pair sequences that we refer to as codons. So AUG is a codon, GGG is a codon, CUC is a codon, AGC is a codon, and GAC is a separate codon. Each of these codons, through the genetic code, which will be discussed in Chapter 4, each of these 3 base pairs is going to dictate a separate amino acid, which will be incorporated into a growing protein. So, for example, AUG encodes the amino acid methionine, whereas GGG encodes glycine. CUC encodes leucine, AGC encodes serine, and GAC encodes aspartic acid. Do you have to memorize all those? Absolutely not. If you're doing that, that's not efficient use of your time, okay? So you will always be provided with a genetic code table if asked to provide amino acid sequence from a given messenger RNA. Why do they call it the coding strand? Because as we've defined here, it looks an awful lot like the codons in the messenger RNA. The key difference being there's U's substituted for the T's. Because you don't find T's in mRNA, and by the same token, you don't find U's in DNA. So for that reason, you would have the T's substituted with U's. Another thing that you should consider is a classic board question is to give you the sequence of the coding strand of DNA, or give you the sequence of the template strand of DNA, and ask you what is the mRNA from this going to look like. Classic question. Or to give you the mRNA sequence and ask, what is the DNA that was derived from, what does it look like? So you need to know what the coding strand would look like, and you need to know what the template strand would look like. So just to practice this, there's some sample questions. Okay, so the following. During RNA synthesis, the DNA template sequence TAGC would be transcribed to produce which of the following sequences? Okay, let's break this question down because it's very likely you'll get this type of question on your exam. During RNA synthesis, the DNA template sequence TAGC would be transcribed to produce which of the following sequences? Now, I know that many listening to this and watching this are wondering, well, wait a minute. You're telling me the sequence is TAGC, but how do I know which end is the 5' and which end is the 3'? How do I know that? Well, if you're not given specific direction as to which end is the 5' end and which is the end is the 3' end, guess what you're supposed to do? You are supposed to assume that the left-hand side is the 5' end and the right-hand side is the 3' end. Okay? So for this particular answer, the answer is E. Why is the answer E? Let's go through and explain why. So when giving one of these types of questions, the way you need to approach it is to look at the sequence and draw in which end is the 5' and which end is the 3' end. Okay, you need to draw in which is the 5' and which is the 3' end, and having done that, you can now approach answering this question in a logical way. Now in this case, the question is asking about RNA synthesis. So what would the product look like in RNA synthesis? Would it have any T's in it? Absolutely not. So right away, you could cross out any option with a T, right? Because you know RNA has U's and not T's. So it's RNA synthesis, so right away, we can cross out any option with a T, and we're left with, just like every good medical student does, or person taking an exam, they're left with 2 answer choices. So at least you have a 50-50 shot, right? So reach in your pocket and flip a coin, right? No, we want to make it even easier for you to understand, for you to answer this question correctly. So, how do you know whether the answer is D or E? Well, if you chose D, you were simply forgetting about the rules of DNA synthesis and also RNA transcription. Remember, what you're going to do when you're transcribing a DNA template is which direction are you going to read it in? You're going to read it from 3 to 5 prime. So as you read this template from 3 to 5 prime, you're going to synthesize a product complementary to that in the 5 to 3 prime direction. Something like this. As this template is read from 3 to 5 prime, CGAT, a product is going to be synthesized from 5 to 3 prime in the complementary fashion, which will read GCUA from 5 to 3 prime. So many probably looked right at choice D and said, aha, this reads AUCG. That's the correct answer, right? Well, at first glance, yes, seemingly that looks like the correct answer choice. But over here, which direction are these being reported in? What's the default? What's the convention in which these are being written? It's 5 to 3 prime. So all of these answer choices, you're supposed to infer that all these answer choices are being given in the 5 to 3 prime direction. So this sequence up here, although from 3 to 5 prime reads AUCG, notice that from 5 to 3 prime it reads GCUA, making choice E the right answer choice. So in summary, to approach this type of question, what I want you to do is first, write out which end is 5 prime and which end is 3 prime. You'll have a little laminated sheet of white paper next to you with some dry erase pens, and you'll be able to do this. The next thing I want you to do is to pretend, if you're being asked about DNA synthesis, pretend that you are DNA polymerase. If you're being asked about RNA synthesis, I want you to pretend that you're RNA polymerase, and immediately look for the 3 prime end. Look for the 3 prime end and draw in the complementary bases going from 3 to 5 prime. But you know that replication and transcription are anti-parallel, so really associated with the 3 prime end of the DNA is the 5 prime end of the mRNA. In that way, you will get a product, you'll know which end's 5 to 3 prime, and you can immediately go match that up with one of your answer choices. So for another example, the following question. Transcription of the following sequence of the tryptophan operon occurs in the direction indicated by the arrow. What would be the base sequence of the mRNA produced? In this particular example, we're being kind to you. We're telling you that transcription happens from left to right, as it's written on your screen. Now, in reality on the test, they might put the promoter and the terminator and expect you to know that transcription always goes from the promoter to the termination sequence. So how are you going to approach this? Well, you know that if transcription's happening from left to right, which strand is the template strand? It's the strand that reads from 3 to 5 prime in the same direction that the transcription is happening. So in order to answer this, you need only read the template strand from 3 to 5 prime and to put in complementary bases, but in the 5 to 3 prime direction. Well, the nice thing is this has already been done for you because they've given you the coding strand. So in order to answer this question, all you really need to do is to take the coding strand, identical sequences, except replace the T's with U's, and that will give you the correct answer. Okay, GCA, GTCG. The correct answer is the same, GCA, but it's GUCG. Right? It's the same as the coding strand here, except the T's have been replaced with U's. So if you're given the template strand, it's very easy. Use the same sequence, just replace the T's with U's, and you're there. If they only give you the template strand, you're going to need to put in complementary anti-parallel sequence in. Okay, so where there's a C, there's going to be a G. Where there's a G, there's a C. Where there's a T, there's an A, etc., 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 going to the end. Where there's a C, there's a G. You should be very prepared before your test day to answer this type of question because I think the likelihood of this being on your test is extremely high.